Hi, so we'll talk about COVID-19 in this video. We'll address four different topics here. So first we'll try to understand what is the cause of the disease. Then we'll look at the symptoms and effects on the body. We'll also try to understand how the disease is actually detected, and we'll try to understand how the disease is actually treated. So these are the four questions that we'll address. Before that, we'll talk about some basics, especially we're talking about bacteria and viruses here. So there is one common point that you need to understand. For more than 3.5 billion years of Earth's history, only unicellular organisms were the norm. Multicellular organisms did not really exist. So they still rule the world, and it is estimated that for every human cell in your body, there are 3 bacteria cells in your body. So that is one estimate. So they still just about exist everywhere, and they dominate your own functioning. So there's about one basic point. About bacteria, they are prokaryotic, which means their nucleus is not really having an envelope. They are of variety of shapes, so they can be of any shape from spherical shape to rod shape. They're usually sized in micrometers, and they are among the first forms of life to have evolved in this particular planet. So they also exist everywhere. They are essential for most animals. They manufacture something called vitamin B12, which is required for most animals. So bacteria manufacture that, and then they also have important functions in human beings. So they are very important for the human digestive system, and the human beings actively use bacteria for a lot of purposes also. Some of them are of course pathogenic. So most of the bacteria that exist in our body are harmless, or they have some positive effect. Some of them of course have some sort of a negative effect. So the body tries to counter that, and that is called pathogenic. So there are some examples of diseases given here. So you have cholera, leprosy, tuberculosis, and plague. So cholera is something that we are still struggling with in some places. Tuberculosis is also one major disease that we are struggling with. Leprosy, to some extent, we have controlled. Plague is something that afflicted large populations over in history. So that was one major disease. About viruses, the most important feature, one of the important features is that it is submicroscopic, which means you cannot really use a microscope to look at a virus. Sometimes viruses are 1/100th the size of bacteria. The most important feature is that they replicate only inside the living cells of an organism. So this is something that you need to remember about virus. Outside a living cell of any organism, it is considered to be non-living. At the risk of oversimplifying, it is considered to be non-living, and they are basically just strands of DNA or RNA encapsulated with some protective layer. And when they come into the organism, they replicate very quickly. So that's one thing. They infect all types of life forms, including other microorganisms. You might have heard of a particular virus called bacteriophage, which actually specifically target bacteria. So they are very numerous. They are found just about everywhere. This is something similar with bacteria. Both of these microorganisms have been very successful over the years, and they are just about everywhere. So it should come as no surprise that there are some types of viruses and bacteria for which we do not really have the immunity power. So obviously that causes some sort of dis-ease. So we have HIV, for example, in virus. We have still not found a cure for HIV. It does not allow the immune system to actually find a way to counter it, because it keeps on changing itself. We have hepatitis. We are struggling with hepatitis again. We have eradicated polio to a large extent, but it is coming back in some countries. The coronavirus is an ongoing pandemic. It is a family of viruses which include the SARS, MERS, and the COVID-19. We are also struggling to some extent with influenza. So these are some examples of virus-caused diseases. Coronavirus. So coronavirus family is basically a family of RNA viruses. So like I mentioned before, viruses are basically strands of DNA or RNA encapsulated within some protective layer. So coronavirus is basically an RNA virus, and it infects birds and mammals, and it causes respiratory tract infections in birds and mammals. Typically, it has club-shaped spikes that project from roughly spherical surface. So these are some basic features. We have identified 6 human coronaviruses. Human coronavirus refers to the coronavirus which actually infect human beings, and 4 of them are known to be mild. They have very minimum symptoms in human beings, while 3 are potentially severe. So one is MERS, which refers to Middle East respiratory syndrome. It was a major outbreak. The other is SARS, which is severe acute respiratory syndrome. It was a major epidemic in multiple parts of the world. 800 people died in the most recent epidemic. And then you have COVID-19, which is an ongoing pandemic. So COVID-19 is already known to have two different strains. So one strain spreads more rapidly, and the other strain produces more viruses, produces more copies of itself. So that is about the two strains of coronavirus. And they are the word mentioned here is at least. So we don't really know how the coronavirus is mutating. It's possible that more strains would actually emerge. So coronavirus is not something that affects only human beings. It affects a lot of animals. The most common word that you would have heard is swine flu. So that is one major disease. It also affects cattle, and it has killed a lot of domesticated animals. It also affects our domestic pets, including cats and dogs. Birds, domesticated birds are the industry is very severely affected by this particular disease. Bats are supposed to be natural reservoirs of the coronavirus family, including probably the COVID-19 disease. Pangolins have been in news because it is suspected that they were the intermediary between bats and human beings in actually transmitting the virus to us. So this is some basic features of the coronavirus family. We'll talk specifically about the COVID-19 disease and the effect it has on the human body. It is caused by SARS-CoV-2. So technically, COVID-19 refers to the disease, and SARS-CoV-2 is actually the virus. So there are a wide range of symptoms. So these symptoms are of course common with other types of diseases. You have fever, cough, and fatigue. So these are very standard symptoms. Specifically for coronavirus, breathing difficulties will be there, and you also have loss of smell and taste. It is said that more than 80% of the people who are infected with COVID actually experience loss of smell and taste. Many people do experience breathing difficulties, and symptoms begin 1 to 14 days after infection. So that means initially the person gets the COVID-19 virus, the SARS-CoV-2 virus, and then the virus multiplies itself, and only after some amount of multiplication, the body exhibits some symptoms. So that is one thing. So there are some numbers mentioned here which are not really accurate. They have been taken from April 2020 data. So according to April 2020 data, you have 81% mild cases. This is globally. So 81% mild cases. 14% of the cases are severe and 5% are critical. So mild cases will basically mean some slight cold or some slight fever, for example. Severe cases will basically mean you have very heavy breathing difficulty. Critical symptoms might include heart attack, might include other complications in the body, and multiple organ failure. So at least 1/3 of all the cases are asymptomatic. Again, according to April 2020 data. Now the data will vary. We do not really know the number of cases which are asymptomatic. So the basic point here is that COVID-19 affects the human body. The person can be infected with the virus. He can actually transmit the virus to someone else, but the symptom need not really come up in the body. Long COVID is also something that you would have heard about. So long COVID refers to the feature of COVID-19 where the effect is actually prolonged over a very long amount of time. So probably the virus is not really traced in a subsequent test, but the effect is still there for a lot of people. So this is not something that we understand much because it's only been 1 year after COVID-19. So more research is going into that. Transmission of COVID-19, basically it transmits between people who are in close contact. Small droplets and aerosols through the air will actually transmit the virus. So it is important that you keep wearing masks and have some social distancing. So that is the rationale behind that. What is the infection process? The COVID-19 enters the nose and throat and finds the nose to be a very comfortable home. Nose and the respiratory tract to be a very comfortable home. That is basically because there is some receptor called as ACE2. So the name of the receptor is ACE2. So the ACE2 receptor is responsible for controlling blood pressure in cells. So wherever that receptor is present, the COVID-19 vaccine is very easily able to bind with that particular cell. So it will bind with the host cell and it enters the cell, it injects itself into the cell, and then it hijacks the cell's machinery and makes copies of itself. That's how most microorganisms function. And then eventually, if the immune system is not able to suppress the initial inflammation, then it will enter the alveoli of lungs. So alveoli is a small balloon-like structure in lungs, which is responsible for the transfer of oxygen and carbon dioxide between the blood and the lungs. So that particular portion of the lungs will be affected, and it is very rich in ACE2. So the coronavirus actually likes being there, and it can multiply very easily. So cough and cold are ways to expel the virus from the body. So basically, this is something that happens for most of the diseases. You cough a lot, you exhibit fever, and you have cold. So these are ways in which the body expels the virus. But unfortunately, in the process of expelling the virus, you actually transmit that to someone else. So this is how COVID-19 infects, and these are some of the symptoms. So here in this slide, we'll get a picture of how bad the COVID-19 can really be in some cases, especially in severe and critical cases. The most important organ that gets affected is the lungs, of course, because it is a respiratory tract infection. So lungs are ground zero for the battle between the body and the coronavirus. You have blood clotting. So these are some of the symptoms which are reported quite often. Blood clotting is reported after, in autopsies. In autopsies, some other symptoms are also reported widely. Excessive immune response is also mentioned. So initially, the first stage of the virus coming into your body is that it infects some of the cells, it quickly multiplies itself, and the immune system is deployed by your body, and it has its effect. But in the process of destroying the virus, the immune system might actually overreact. So that is referred to as cytokine storm. So cytokine is one particular molecule that is more like a signaling mechanism to bring in more immune response. So that is about excessive immune response. Excessive immune response might actually make a person die. Hypoxia is one very common feature for COVID-19. Even if you are infected with COVID-19 and do not have breathing difficulties, hypoxia is something that is very common. So you can test the oxygen level in your blood, and if you're having COVID-19, the oxygen level will be very low, as seen in the oximeter. Brain inflammation, stroke, and seizure. It is not commonly mentioned, but it is reported in some cases, in extreme cases. Inflammation in the eye is also not commonly reported, but it is there in extreme cases. These are probably the most important symptoms, most easily noticeable symptoms for COVID-19. You have a loss of smell and you have a loss of taste. It is basically because the virus has its makes the respiratory tract its home, and then eventually when it spreads itself from that place, it might actually reach the nerve endings in nose and might damage the cells, and subsequently you have a loss of smell and a loss of taste. Immune cells will crowd inflamed alveolus. So alveolus is again the balloon-like structure in the lungs, which is responsible for oxygen transfer. So if it is also rich in that particular receptor that we talked about. So the diminishing oxygen uptake is very important feature here. So once the alveolus is inflamed, then oxygen uptake will be affected. So people may die because of that. From autopsy, people have also found that it might affect liver. So enzyme levels have signal that the liver is actually struggling. So that is also reported in some cases. Kidney damage was reported often. So either the kidney is affected directly or indirectly. Directly would mean that the COVID-19 vaccine, the particular virus, it actually attacks the cells of the kidney. Indirectly would mean that because of diminished oxygen level and low blood pressure, kidney does not function properly. In extreme cases, for people with comorbidities, for example, diabetes, heart attacks have been reported. So COVID-19 virus, it clogs the lungs, and eventually the heart does not really function properly, and blood clots are reported. And because of that, eventually it leads to heart attacks. So these are some of the common things that COVID-19 does. So a lot of people have actually died in other countries. So it is important that you still remain vigilant with the COVID-19 pandemic. We'll talk about the treatment for all these diseases. So treatment refers to what you do after you contract the disease. So the COVID-19 vaccine does not really come under this. It's more about the preventive measure sort of thing. So what is the first thing that we have done for treatment? We've tried to repurpose different drugs that already exist. So how do you do that? You first try to understand the interactions between the human cells and the viral protein. And after understanding those interactions, you could possibly use some other drug which will target those interactions and disrupt those interactions. So some of those drugs which were tested were hydroxychloroquine, dexamethasone, favipiravir, lopinavir, and remdesivir. So you would have especially heard of hydroxychloroquine and remdesivir. So these were quite often in news. But in a trial called as a solidarity trial, they have found that these drugs do not really have a large effect on the human body. They have little to no effect. We've also tried plasma therapy. So plasma therapy is where you take the blood of a person who's actually cured with COVID-19. You take that blood and you transfuse that into the infected person, the other infected person, so that the antibodies in that blood might actually help the other person develop an immune response. So that is about plasma therapy. People are not really confident about plasma therapy because it does involve some amount of risk, but in some cases it has actually helped. So that is ongoing. We also talked about antibodies from llama. So this is one example of a treatment which was not really common, not really talked about quite often, but it was discussed in scientific circles. So llama is one animal which is found in South America. So the llama is actually having antibodies for COVID-19, for this particular coronavirus family. So here the antibodies are taken from llama and then they are injected into the human body so that the body will actually generate an immune response. But for most of the time, for COVID-19, symptomatic treatment is the norm. So if you're infected with COVID-19, you're taken to a hospital and based on your symptoms, some sort of medication and treatment is given. So that is the thing that most of the people get. We also try to suppress immune system response at, in the later stages of the infection. In the initial stages, you do not really do this. In the later stages, if the immune response is very high and if it is actually causing harm to the body, then you could use drugs which will reduce that particular response. So these are some things which were mentioned in news. You might remember this. Please do not try to inject bleach into your body because that is an indication that you will lose the election. So you could also try to avoid drinking ethanol. So there were a lot of cases where people in India, because of the lockdown, they actually stopped. They actually could not consume alcohol and they went for hand sanitizers. So that caused a lot of death. So that is also something that you could avoid. Yeah. So we'll talk about testing technology in this slide. So there are a lot of methods to actually test for the presence of COVID-19. So there are basically two methods. First, you try to detect if the virus is actually present in your body, or the other thing that you could do is see if antibodies for the particular virus is actually present in your body. So you could do these two things. So we have this most important test called RT-PCR. So the expansion of that is reverse transcription polymerase chain reaction. So this is the gold standard for testing. So this is something that takes quite a lot of time. It has its share of disadvantages, but it is very accurate. So reverse transcriptase, this part refers to the conversion of the RNA of the particular virus into DNA, and then polymerase chain reaction refers to the process which actually multiplies that DNA to a very large number so that it can actually be analyzed. So first you convert RNA into DNA and then you multiply it to a significant number so that you can analyze it. So based on the analysis, you can find out if the particular individual is infected with COVID-19 or not. So this is the gold standard for testing. So this is used in quite a lot of places. It takes quite a lot of time. That is the most important disadvantage. This is real-time PCR. Real-time PCR is also called qPCR. The significant advantage here is that it is quicker, it does not take as much time, and the process here is very similar. So that is one major thing. So these are examples of testing where you are actually testing for the presence of the virus in the body. So how do you sample this? So you have different ways to actually get the sample of the coronavirus. You have you can take samples from the nasal area, or you can take samples from the pharynx. You can also take sputum, throat swabs, and saliva. So in most of the cases, you take this nasopharyngeal swab. In some cases, you can take sputum, but what sort of sample you take is very important for the accuracy of your test. It is shown that if you take sputum, the results are actually more accurate than if you take nasopharyngeal swab, but this is taken most of the time. Some tests are there which actually use saliva. It is comfortable both for the person who is testing and also for the person who is getting tested. So you also have throat swab. So these are different ways in which samples are taken, and then it could be used either in RT-PCR or qPCR testing. There are other testing methods like isothermal nucleic acid amplification. So we are talking about isothermal here, which refers to the temperature being constant. In the RT-PCR test, actually the temperature is varied in cycles. It's part of the process, but here that is a major disadvantage because it requires a lot of infrastructure. But here it is kept isothermal and it's basically nucleic acid amplification. And the most important feature is that you use CRISPR technology for this. So under this particular test, there is a particular enzyme which targets the protein, targets the DNA. Targets the RNA of the virus, and then it binds with the RNA, and some sort of color coding is done so that you will actually get the test result. So that is about isothermal nucleic acid amplification. So this is the other thing that you do, antigen testing. So this is part 2. So this is about part 1, this is part 2. So antigen testing, you can test it even before the onset of symptoms. So that is one thing that you can do. You also have sniff test. So sniff test basically means you objectively try to determine if the person is able to smell or not, because loss of smell is a major symptom of COVID-19. So you could do that. CT scan of chest is also done. It was started in China initially, but it lacks specificity. So specificity and sensitivity are two terms. Sensitivity refers to the percentage of positive cases which are actually correctly detected. Specificity refers to the percentage of negative cases which are correctly detected. So CT scan of chest will definitely show you the positive cases, but it is not really good enough to show you the negative cases. It cannot really say if a particular case is negative. So that is very low there. For antibody tests, on the other hand, the specificity is very high. So antibody test is basically this other part where you actually test if antibodies are present in the human body after the infection. So to be very clear, antibody is the part of the immune system which actually it's part of your body which actually fights the virus. So antibody presence will mean that the body has actually been infected with COVID-19. So you can definitely say that some particular person is not really having COVID if antibodies are not present. So therefore it has high specificity, but it has low sensitivity. So that is one major disadvantage. So there are a lot of different tests here under antibody tests. You have rapid detection tests, you have enzyme linked immunosorbent assay, it's also called as the ELISA test. You have neutralization assay, chemiluminescent immunoassay, etc. So a lot of different testing methodologies have been created by different institutions. So that is about the basic testing. So the most important point here will be RT-PCR because it is the gold standard. You also have qPCR because it's faster, and the tests here are antibody tests. It is more for the purpose of purpose of the government trying to understand how much of the population has been infected. But this is something you use to actually find out if you are infected in the present. So that is about testing on the whole. So the testing methodology also varies quite a lot. You could choose to test in different ways. So this is one thing that is common. It's probably available everywhere. So if a particular individual wants to get tested, testing centers are available for him to go to and actually get tested for coronavirus after paying some money. So home collection of samples is also there, but it's not necessarily after paying money. You also had COVID-19 camps where free testing was done. So home collection of samples, this model was conducted in New Delhi. So New Delhi, the coronavirus cases were very high. So the government decided to actually go to individual homes and actually collect samples for testing. So that is something that you could do. Pool testing is a concept where the samples of a lot of individuals are put together and then the entire sample is tested for COVID-19. So that is used if you really want to understand if a particular locality or if a particular family has been infected with the virus or not. So that is about pool testing. So these are some different types, but the focus of testing can also be different for different people. So the government the regulatory bodies could try to focus more on people with travel history because that was initially one major deal. In China, if people travel to China or people travel to Europe in the initial phase, then they were tested more aggressively than the other population. So you could also test people with symptoms because symptoms are a major indicator of there being some problem in the body. So you could also choose to test people with symptoms. People in containment zones could also be tested. So containment zones are places where the number of cases are very high. So naturally, the possibility of more cases coming up is also high. So you could choose to test there. There are some people who are involved in essential services. You have frontline workers, including doctors, nurses, and also policemen. So these workers are constantly in contact with people, so they could also be tested. And people who come up in contact tracing, so that is also one major group that you can possibly test. So the major point here is that in the different stages of the pandemic, different groups of people are actually chosen and they are tested accordingly. So this is about the testing methodology of COVID-19. So this has basically been a short video talking about COVID-19 and how it affects the individual. So we talked about the different symptoms of COVID-19. We talked about how treatment is done for COVID-19. We'll probably try to cover vaccines in a different video. We also try to understand the different testing methods and the testing methodologies. So that will be the end of the video. Thank you.